Welcome. I'm Dr. Timothy Murray for the Foundation of the American Society of Retina Specialists, bringing you our Milestones in Retina. It's a real pleasure and an honor to introduce my friend and colleague, Dr. Mark Humayun from the USC Roski Vision Center, who has been the director and co-inventor of the artificial retinal prosthesis. Mark, we're really looking forward to your telling us the journey that you've taken over almost 3 decades to bring your first idea into practical delivery for our patients, and then to tell us where that future is going to lead. So thank you so much for joining us today.
Happy to be with you, Tim. I'm really looking forward to this.
So Mark, for a start here, tell us what brought you into the idea of vision restoration, and what was unique, do you think, early in your career that allowed you to be so focused on this?
Well, I wasn't interested in ophthalmology or vision restoration in medical school, but at that time, my grandmother started to go blind from complications of diabetic retinopathy, and this was so long ago, we didn't even know focal laser would actually work. We knew pan-retinal photocoagulation was working for proliferative disease, but she had a non-proliferative, severe non-proliferative, and we didn't know. So this was that long ago, and I still remember taking her to, the Wilmerines to see to Scapins and Duke and other places. But it was very, very frustrating. And so it made me focus in on ophthalmology, in particular retina and retinal blindness and what could be done. And at that point, I had heard about cochlear implants. There was a lot of excitement. I was at Duke. And so the thought came, well, could we actually, implant something like a cochlear implant for the eye? And at Duke University Medical School, there's this third year you get to spend it away or at Duke doing research. And I was fortunate enough to spend it at Johns Hopkins, where in neurosurgery, I saw a patient being stimulated. It was for epilepsy. They were trying to resect certain parts of her epileptic brain, but when they stimulated the visual cortex, the patient said they saw a spot of light. It was just amazing to me. There was no light in front of the patient, but she could see it and very reliably tell where it was. So it was a combination of seeing that at Hopkins in neurosurgery, and the combination of hearing about cochlear implants at Duke, and of course the motivation from my grandmother that sort of triangulated and made me focus on potentially, building a retinal implant.
But in addition to your MD, you pursued a PhD in engineering, which gave you a unique focus almost to look at this, different from what many other people would have the opportunity to experience. What was it like getting your PhD?
Well, I tell you, it's not, when I looked at it, I wanted to get a PhD in anything but engineering. And the reason was not because I didn't like engineering, I liked it. But I needed to take 56 credit hours because only 4 credit hours of physiology would transfer, versus if I, got a PhD in just about anything else, neuroscience, etc. It would be only, 25 credit hours. But all that to say, at the end of the day, I realized back then, Dr. Mockemer was the chairman at Duke, and so everybody wanted to work with Dr. Mockemer, and Diane Hatchell was there too, and everybody wanted to work with her, but really nobody wanted to work with Jean Dewan. So, and so I approached him, and he was working on angiogenesis back then. Judah Folkman, of course, we know the story, and so I suggested this idea, and he had just invented a retinal tack. So he kind of liked the idea of putting something in the eye that was foreign on the retina. So he allowed me to run with it. And I soon realized that one of us needed to get an engineering degree, and it wasn't going to be Gene. So I ended up doing it, getting my PhD in engineering, just because it was literally, I would sit across engineers and I would try to explain and it was like talking a different language. I mean, they would tell me, explain me sodium, potassium, and plasma membrane, and similarly, I didn't understand a transistor. So we just weren't getting anywhere, and so getting that PhD really in this area in particular really allowed me to be able to speak both languages, and I like it. So I liked engineering. It wasn't that it was that I didn't like it. It was so brutal to look at 50. I mean, and there were 6 math courses that had to be taken one after the other. So so it's an interesting time, but that sort of gives you a little bit of a backdrop to it.
Well, you certainly paid your dues to get your foot in the door from the MD PhD side for that. But I would venture that just like when I got my MBA and it changed the language discussing patient care with an administrator, it allowed me to really speak to them in the way they needed to hear what I was saying. So it sounds to me like that was a big step for you also, to be able to speak that language. So you put a team together, and what was the first approach? Because you have an implantable chip, you have cabling lead, you have power supply, you have visual processing, you have software. Can you take us a little bit through that development step that gets you to the Argus One? And can you tell us the first moment when you really thought there was some feasibility for this approach from a retina standpoint?
Well, we started working on frogs, frog retina. It's easy to maintain. It's amphibian, you could imagine. There's some interesting stories about frogs getting loose in the basement. I was always relegated to the basement, so that's where all the labs were, and every time, whether it was at Duke or Hopkins later. So fortunately, with the frog did get out, it was in the basement. But having said that, a lot of frog stimulation, but really things changed when we did the first short-term test in a blind person who had X-linked RP, had been blind, totally blind, for nearly 50 years. As we know, X-linked is more aggressive, and so by 22 he was pretty much had lost all his vision, and now he was in his early 70s. So 50 years later, we put 2 single electrodes through the pars plana, 20 gauge incision, and we only anesthetized the area of the pars plana because we couldn't block the optic nerve, and stimulated. And that was really the time where things changed, because this was the first time the patient, a blind person, actually saw a spot of light in response to controlled electrical stimulation. And of course it wasn't that simple, because for the first 20 minutes nothing happened. And we looked at every single circuit and every little wire, and making sure that everything was connected properly. And then the patient tells us that, oh, you see this little tiny candle flickering at an arm's length away. And we knew that's what it was. He just hadn't seen it in a long time, so one would anticipate that it would take him some time to understand or even appreciate the visual perception. But we knew that he was seeing the electrical pulse. Now, electrically generated visual perceptions are these technical things called phosphenes. So we knew we could get him to see the phosphene because we would turn up or down the frequency and ask him to count. And if he could count along the frequency of stimulation, we knew that it was a phosphene. So that really changed everything. And from there on, we had to build a permanent implant. And you could imagine, attaching something to the retina. I still remember presenting this to Dr. Makamer, and I thought maybe I just completely had gotten the wrong idea, because he's like, he basically was teaching me. He didn't say son, but something like that. I mean, he was very formal, but he said, the retina is very delicate. What are you talking about? Attaching something to it permanently. So all that to say is, after that, to Dr. Makamer's credit, he loved talking about this project, and no matter where we were, at Gonan, where he would be surrounded by everyone, or whichever society he would be, he would always come find me and sit me down in a corner and insist for talking for 30 or 45 minutes about the project. So he was always very, very interested and very supportive. But that first meeting was obviously pretty rough, trying to tell Dr. Markham we're going to attach electrode array to the retina and not have it detach. Then this whole idea, so all that to say is the surgical procedure had to be retooled and rethought about. Putting a cable across the sclera. I mean, Tim, you know that. I mean, I'll let you comment on that. We put tubes into the pars plana occasionally, mostly into the anterior chambers. So that was tough. And how do you suture that? Then, of course, tacking electrode array. How do you do that? Without destroying the retina and detaching the retina. And so a lot of surgical challenges. But we could talk a little bit more about the other components.
Before you move on to that, I've always thought that your approach using an epiretinal array, as opposed to attempting to place a subretinal array, was really a remarkably novel and thoughtful approach to do this. So I know would have looked at all the different variables to how to do this. What drove you to an epiretinal array?
Well, really good question. There weren't, it was either subretinal. We thought about, suprachoroidal, but or episcleral. So there was a project back at Duke where they were making this glove for Wolff Parkinson White reentry arrhythmias to the heart. So it was a glove, and so we pictured, well, could we put a glove electrode outside the sclera and have it function? But it would stimulate the rectus muscles too much, and twitching would occur, and you'd be too far from the target. Similarly, suprachoroidal implants. I know some groups are still pursuing that, but it just was again too far from the target neuron. So we got subretinal. It was challenging because you would have to go across the choroid, which of course others have done, or put something transvitreally across a retinotomy. But you can't have a cable going across the retinotomy. Either you do a completely self-containing solar, which is some companies did that. Either you do that, or the moment you have a cable across the retinotomy, it doesn't work. So all this thought process led into developing something epiretinally. But of course, there are the axons, and so the first thing you encounter are the axons, and how do you not stimulate the axons? And that's where some of the very sophisticated work that a number of my students, including Rob Greenberg, who went on to be the CEO of Second Sight, and others, we looked at that, and it turns out that the axons are 1 micron and they're unmyelinated, so they're very difficult, unlike myelinated axons, which have nodes of Ranvier, where currents sort of collect and go in and can depolarize. Single fiber, thin, unmyelinated axons are difficult to stimulate. So we got lucky there, and so we could actually stimulate soma. So all that to say is a lot of thought process, and we also ended up, one last thing, we ended up putting the electronics outside. There's too much thermal dissipation, and yes, we have the vitreous cavity, which is 3 cc of water, which is a great heat dissipator, but when the electronics are touching close to the retina, there's too much dissipation of heat. So putting the electronics outside, putting the cool, non-thermal, aspects of the device inside, and then tacking it seemed to, in the end, after all this analysis, came out to be the way to go, provided we didn't accidentally stimulate a lot of those axons.
So a lot of thought and a little luck goes a long way, so that's key. And you clearly put together such an amazing team to do that. So the thought process gets you there. You know that you've got a proof of principle. Take us through the journey to get a first prototype device. What were the biggest challenges for you early?
Well, there were so many challenges, but we're probably the challenge was hermetic packaging. Hermetic packaging, it's a technical term, but for these implantable electronic devices, they have to be resistant to water vapor. And actually, the FDA uses what's called a helium. Helium is a smaller molecule leak test. So these packaging for the electronics have to be helium resistant, and so to like 10 to the minus 11th. So, very, very small amounts. And so that's very challenging to make a package. And every wire that comes out of a package is a source where it could leak, right? And so we said, okay, big challenge, have to make a chip, all this stuff. So what we'll do is we'll take proof of concept. We're going to hijack a cochlear implant. And everybody looked at us and said, what? And we said, we're going to hijack a cochlear implant, 16 electrodes, 1/6, and we're going to put it where it goes normally for cochlear implants, but then cable underneath the temporalis muscle go around the orbital rim, and then suture the cable and put it into the eye. So that was the procedure. We did hijack so to speak, the cochlear implant, and we made it so you could now, instead of having to, you could receive input from a camera on the glasses. And that was the Argus One. The Argus One was 16 electrodes, and everybody said, 16 is not going to do anything. But for us, it was the quickest way to get a long-term implant. So the Argus One was a hijacked cochlear implant with 16 electrodes, and we did 6 subjects with it, and it showed that what it showed, the most important thing which we were looking for, is over a long term patients could actually see form vision. They could actually put the dots together to see large letters and objects, and that the thresholds and the currents required were very stable, and we didn't damage the eye, and the eye didn't damage the implant.
It's amazing to think about all the unknowns here. I mean, and it's amazing too that you had the insight and the foresight to take an existing implant and transfer some of that technology to apply to the eye. But in many ways, very unique. So you have to have a visual processing unit. You have to link them. You have to think about battery sources. I think the cochlear implant, in so many ways, I don't want to be disingenuous, but it's relatively easy in my mind compared to what you were trying to do. So how did you work your way through the visual processing, the camera systems, the software, even thinking about battery? And Argus One started, but you made some huge advances from Argus One to Argus Two.
Well, I think those are exactly the areas we struggled with, and Argus 2, the Argus 1 was too difficult a surgery. It took 7 hours, and cochlear implant companies were not interested in developing a retinal implant, so they weren't going to let us do it too much beyond a test device. So we had to build our own device. We had to make it such that vitreoretinal surgeons could implant it without the help of plastics and ENT surgeons and so forth. And we had to make more electrodes. So this led to, the challenge is the hermetic packaging, which now we have 60 wires coming out of this little package. The package had to all of a sudden been shrunk from, 1 inch to 10 millimeters. So dramatic 2.54, to down to 1 or so centimeters, that is. And so at this point, you have more electrodes coming out, smaller body. So the cochlear implant technology just didn't support it. So it had to come up with new packaging schemes to do that, to get the hermetic sealed. And of course, the new integrated chip, we call the ASIC, it's called the application specific integrated chip. Actually, it's a mixed mode chip, to your point exactly. It receives telemetry, so it's wireless, but it has an analog circuit because it has to put out currents in analog, and it has digital circuitry to process. So it's called a mixed chip because it has telemetry, analog, and digital circuitry. So these chips are very complicated to make. And then, of course, the electrodes. The cochlear implants electrodes were handmade. They are individual, discrete platinum wires that are put into silicone rubber. And we can't do that for 60 electrodes. So for the very first time, we had to use principles of photolithography, which is how you make integrated chips. But instead of making the chip, we had to actually now deposit the metal lines onto the plastic polymer and make it all flexible and foldable. So that photolithography and electrode array took some time. And then to your point, even the camera was fortunately something that was they were much more advanced, so we didn't need as advanced a camera, but we had to figure out the visual processing, and that's the video processing unit, because you can't, be 3 seconds late, somebody crossing the street with a bus coming, obviously that would have bad outcomes. So real-time video processing had to be developed, and so we had to segment the image, even 1 megapixel, and how to handle it, how to handle 256 grayscale levels, incredible amount of complexity to it. And then what we had to do was basically make sure that all this information was conveyed to the retina, and that perhaps was the hardest part. I mean, you would think all this sounds hard, which it was, and so now the hardest part is, okay, what pulse? How do you shape it, and how do you stimulate the retina to get the best effect? And you would think, well, okay, I present one and have the patient look at it and say, better 1 or 2, like we do with glasses, and provide 2 different patterns, and they'll be able to tell us. But if somebody hasn't seen in 30 or 50 years, unfortunately, they tell you 80% of the time wrong. They'll tell you the wrong direction. And probably because there isn't all that much difference anyways in the early, when you're presenting 2. So both those things confounded factors, and we had to use artificial neural networks to converge in spite of the fact that 80% of the answers were wrong. So, it's akin to, I'll share a little bit of a story. It's akin to, you want to, climb Mount Everest, but if you tell the algorithm or the machine never to go down, well, if there are hills and valleys, it'll never make it. So you have to allow the algorithm to go wrong and still converge. So, machine has to go up and down in valleys eventually to get to the top of a mountain. Similarly, we had to use a lot of different algorithms to get there. So all that to say is the software was challenging, hardware incredibly challenging, image processing, and the surgery we've already talked about. So there really wasn't anything easy about it, but I can tell you, it was a lot of fun, and that's what kept us going.
Well, and I think you had that positive feedback from those patients that really appreciated the impact that it had on their lives. And, I think we all were wanting more, but I think we sometimes forget what that bit that you gave them, what it meant to those patients. So I think that's pretty remarkable. And then part of the story seems to me that once you had everything in place, it was also about how to have the patient understand what was this vision. Is that true?
Yes. What was very interesting is we didn't have to do too much of that, but what we had to do was, it wasn't like learning a whole new language, but what it was is we had to train the patients a certain way. And that was really important. If you trained a certain way, our first patient, it took us 2 years to get them to a certain level. But once we understood the training algorithm, we could get there in 2 months. So it's how you train them. And cochlear implants similarly train patients after implants to get them to the level of function they need. So that training is good. And so let me give you an example. Cochlear implants patients do really well because they can lip read and they get another cue. But what could we do? And what I thought what we could do is to have tactile feedback. So, okay, feel this is like a triangle. This is a square. And look at it. And so having that other sensory feedback was very, very helpful. And so I think that, those sorts of things in training help, because the camera's in the glasses, you don't want them to move their eyes around to learn more, to do sort of the head scanning. That takes a little bit. So you have to, if you train them properly, things go very, very well. And that was very important.
take us to the moment that you're putting the first series of the Argus second generation implant in. It seemed like that was a huge step forward in the technology, but you'd already learned a lot through Argus One. So I'm assuming that transition must have been a little bit easier for you and your team and your patients.
The transition was easier. It took 5 years to go from Argus 1 to Argus 2 just to be able to manufacture it. And even at that, as you know, the yields were low, so 1 or 2 out of 10 would make it. But you're right. Once we built it from a surgical standpoint, it was a lot easier. I think everyone had to sort of understand how to put a cable across the sclera and suture around it so we don't get hypotony, how to introduce an electrode array into the eye without, detaching the choroid, these sorts of things, making sure that the pars plana incision is, all the way through the choroid, and then learning how to tack without having a lot of cable effect. If the cable's really torqued and you tack, it may look good at the time, but over a period of time, it sort of works its way against you. So those things had to be done. And then, of course, the hardest thing was to teach all the surgeons that at the end of the procedure, it's not really the end of the procedure, you have to make sure that you cover the implant and close the tenons and conjunctiva. And nobody likes to do that. But if you don't, then you get exposures. And, we've seen that with other pars plana implants too. So that last step is very, very important. But all that to say, you're right. It was easier, and we knew what to expect and how to train the patients. But in some sense, it still had significant, because now we had to, in Argus 1, I was the only one who implanted them. Now in Argus 2, we had to do multi-site, multi-investigator.
And I think taking something, I think it's always interesting when we see our ideas move out of our hands into other people's hands. And I think that that's a stress that unless you've had that experience before, you never kind of appreciate it. So I give you real kudos for how well you've done that. I think you've really trained people around the world to handle that implant well. Now, the next step for us hasn't really occurred yet for our patients, but I think all of us have been looking forward to a next generation implant. So can you give us some idea of what that's going to look like to take us from the start maybe to where we're going to be 3 years from now?
Yeah, Second Sight, was the maker of Argus 2, and I think everybody on this who's listening to this knows that they suspended making the Argus 2, and largely because it's a small company and they had to make a decision whether, they continue with the Retina or do they go forward with the Orion, which is a visual cortical implant. They elected to go with the visual cortical implant. They think it's a bigger market, although I think that the Argus 2, showed already effect, so it was de-risked. So one would think that would be the easier path to go forward. But for whatever reason, I'm not involved in that decision making, but they went with the Orion, and they've implanted 5 or 6 subjects with the visual cortical implant, which very similar to, it's a high voltage Argus device, but very similar. And we'll see what comes of that. So that was a decision made. So it left me thinking, what could we do? We really had just, hit second gear, so to speak, with the retinal implants, but I didn't have the commercial partner. Al Mann, who actually founded Second Sight, was a billionaire, passed away at the age of 90. So things really did change a lot at Second Sight. So I was fortunate enough to partner up with 2 companies, GoldenEye Bionics and IntelliMicro, and they've come together, and we made another implant, which is a 256 electrode. It's called IMIE. That's their term for their new implant, so it's not an Argus 3, but it's an IMIE. And it's been implanted in 5 subjects, the 256 electrode device. It's a single quadrant device, so the antenna and the electronics all compacted, brand new chip, ASIC chip, different telemetry approaches. It has an infrared camera that works with the visual camera to give a thermal signature, so patients don't touch something hot. It has digital zoom capabilities. It has low light capability. It has amazing low light capability. The one lux, like we've seen with Luxturna, it can, subjects can easily move around, objects at that light. So it has all those features, and really looking forward to it. And also, the other thing is that the manufacturing has become much more reliable. So now 9 out of 10 devices are coming offline, working. And so what that translates to is, a lot lower price. And so hopefully that'll make it available for not just the first world, but, everyone, including, people in the third world. So a lot of excitement around the retinal implant. So I'm fortunate that we continue to develop it. It's a 256, but this technology can go to 1024 or even more, 2048. The packaging technology is different. It allows us to be able to do that, to scale up much easier than the Argus 2, Argus 1, or cochlear implant packaging.
So you've looked at theoretical visual acuity functional levels from different packaging pixel densities or pixel light densities. So what do we get with 256, and then where do you want to go?
Well, I think the footprint, if you were to do a visual angle calculation, and these are always very back of the envelope. So what you can do is you can do it that way, and you can say, well, if I have a 60 micron footprint on the retina, what does that project in the field of view? That's one way to do it. Another way to do it is, which is the way sort of what we've done, is you take the Argus and you take the Argus 2 and you take the IMIE. So you have 3 data points and you can draw a curve, and then you can see where things go. And we're doing that, and I think with a 1024, there's no, I think, which is 4 times as much, you could probably be in that range of about 2060 with a little bit of digital zoom. We have digital zoom, so maybe with a 1.5x digital zoom, you could be there at 2060 with those sorts of projections. But a lot has to still go right for us to get there.
I think that what's amazing to me is, you brought some incredible scientific development in multiple aspects of this project. But you've also, been lucky to have things go well at the right time. So I think all of us are excited to see where this would go, because when you're bringing numbers down to that 2060 level or even closer to 2040, that's fundamentally a game changer for literally every retina specialist in the country managing patients. So I find that kind of incredibly exciting. And then to hear that the manufacturing processes now have are so robust that we're seeing good device outcomes for you to allow us to think about reasonable packaging prices. I think that's kind of an amazing opportunity. I'm very excited for my patients. And one of the things that excitement has led me to, and I think you and others, is also to recognize for us today how important preserving anatomic function is in the patients that potentially may be candidates for this in the future. So could you touch a little bit on what we do for those patients and what you think prepares them for this?
Yeah, absolutely right. I mean, we used to put silicone oil in the eye, and I think, I don't know, this was a very long time ago, but I remember one of the talks, maybe it was Dwayne Fuller who gave, he was talking about taxidermy of the eye, and I still vividly remember that term and that talk. But all that to say is, yes, we're not just stuffing or just doing it as taxidermy, but it is really important to save every bit of vision and every bit of neuron and connection from the eye. So 5 patients with macular degeneration with disciform scars, if I think they're going to bleed or they have a huge amount of edema, I do control that. They have high elevated intraocular pressure. I'm very aggressive with them, including patients with RP. So I just, make sure that we don't damage their optic nerve, keep their retina as good a shape as possible. So, future therapies, whether it's a bioelectronic implant, whether it's a stem cell implant, whatever it is that comes along that could restore their sight, whether it's optogenetics, whichever one of these approaches, that there are enough input neurons in the retina for us to work with and restore sight.
So I think that's such a key thing to understand, that what we do now may play a difference in what's available to our patients as you move forward. But having said all of this, I think truly one of the greatest milestones in retina was the work that you have passionately pursued over 3 decades and continue to passionately pursue. So for the American Society of Retina Specialists and for myself, I'd like to thank you, Dr. Mark Humayun, for speaking with us today and for bringing us some unique insight on the development of one of the true milestones in retina. Thank you.
Thank you, Tim.
Thanks, Mark.